Hi, welcome back to part 3 of the lecture video for chapter 10 on muscular system. So like I was saying, we got through most of the difficult part of this chapter, going over all the microscopic structures and the biochemical mechanism of a muscle contraction. Now we focus more on the big picture of muscular contraction, physiology part of it. We talked about and mentioned in part 2 the importance of ATP during muscle contraction. One is for the release of myosin heads from the actin filament, and the other use of ATP was for pumping the calcium ions back in towards the sarcoplasmic reticulum. So the focus now is, how does our muscle fiber acquire their energy? Okay, we already mentioned a few, we already talked about cellular respiration in general biology class. Now sort of, I guess, a review and application of why we need to burn carbohydrates, fats, etc. for producing ATP and what ATP is used for, okay? So hence, in terms of muscle metabolism, there needs to be these things in order for a muscle fiber to function normally. So here is a list of 4 things. First of all, you need substantial intracellular energy reserves. So having enough glucose around so that you can allow for those glucose to be turned into ATP, or sometimes having another type of storage of ATP molecules other than glucose is required, and we'll talk about that for muscle cells. What is that is storing ATP for us? Second, there has to be normal circulatory supply, meaning that you have to have enough blood flow to your muscle fibers, because you need that oxygen to be constantly supplied to muscle fibers in order to use that for cellular respiration and turn the food you eat into energy and ATP to power those muscles. You don't have enough oxygen, there is a backup plan, but still we can't rely on that backup plan too long. Okay, and I'll talk about lactic acid fermentation a little bit when we get to that part. And lastly, your blood pH must be within normal limits, which again goes back to why we cannot rely on anaerobic respiration, your lactic acid fermentation, too long, because then our pH will drop because of the lactic acid buildup, and your muscle cannot go on to doing muscle contraction. Okay? All right. So, let's see. Any changes in levels of these values will reduce the efficiency of muscle activity and also cause premature muscle fatigue. So this is why we have to have constant values of these things. Energy reserve, blood supply, oxygen level, and pH must be always within that narrow range I kind of mentioned in chapter 1 with 7.35 to 7.45. Okay, for that pH. The only energy source our bodies can directly use for muscle contraction is ATP. There is no other substitution, but there can be a number of ways we can generate ATP for our muscle fibers. For example, via something called creatine phosphate. That's way number 1 to generate ATP. Anaerobic metabolism, which is mainly glycolysis, way number 2 to generate ATP, and then aerobic metabolism, which is your citric acid cycle and your electron transport chain, which is way number 3. And then there is a way number 4, which is anaerobic respiration as well, but it is called lactic acid fermentation. That can happen, but we cannot rely on this. This is not for a long-term supply of energy, but just temporary while we are running out of glucose or carbohydrates for energy. Okay, so these are the parts that we can use. Our muscle fibers or muscle cells will rely on for muscle metabolism for generation of ATP. I'm going to skim through glycolysis, citric acid cycle, and electron transport chain very quickly because we already went over that in detail in general biology. Spent 2 chapters or a whole chapter talking about it. So I'm not gonna go into details about those again, but I will talk more about creatine phosphate and what happens with lactic acid after it's generated in your muscle. Okay, so at rest, a skeletal muscle fiber generates more ATP than it needs. So it actually will generate these ATP, but yet, it's resting. Your muscle's not doing much. Just keeping that muscle tone like we were talking about is not using massive amount of ATP to get your muscle cells moving. So hence, instead of wasting those ATP around, there is a way to store them. This is even more faster supply of energy than glucose, because glucose you still have to go through glycolysis, citric acid cycle, and electron transport chain in order to make ATP. But if you have this other thing that is already in ready-made ATP form, and all you have to do is just one step to turn it into ATP, that's a very quick way of generating energy. So, and for muscle fibers, they are able to make, use and store ATP in form of what's called creatine phosphate. And creatine phosphate is just kind of like a little storage box for your extra phosphate group. And when you're ready to use it, it gives up that phosphate group back to your ADP molecules to make ATP. So during muscle contraction, myosin head will break down ATP to hydrolyze ATP into ADP and inorganic phosphate. So the energy stored in creatine phosphate can be used to recharge those ADP molecules. So what happens is, during resting state, excuse me for the noise outside. So in resting state, you have your creatine that can combine with the phosphate group from ATP to make creatine phosphate, CP, leaving your ADP floating around. So now, in a sense, creatine phosphate is serving as a temporary storage thing for those extra phosphate groups, okay? Now, during contraction, now you really need a lot of ATP coming in. So this is why then you can use and quickly gather these creatine phosphate and then remove that phosphate group back to the ADP molecule to give you ATP and creatine right back. And this requires an enzyme called creatine kinase to transfer that phosphate group back to the ATP. And, I hope maybe you remember this from general biology. Anytime an enzyme says kinase, it always involves with movement of a phosphate group from one molecule to another, called phosphorylation. So that's why kinase always has to do with phosphorylation. And in a sense, we're phosphorylating our ADP into ATP. Okay? All right. So, but, and because of this function of muscle fiber, and having this enzyme turns out to be something we can track clinically speaking, in terms of whether we can tell a patient's muscle cells are being damaged. Creatine kinase, technically speaking, are only present in muscle cells. So when muscle cells are damaged, what happens is that these creatine kinase enzyme can leak into the bloodstream. And when we are able to detect that, levels of creatine kinase in a patient's blood sample, then that indicates something serious have probably gone wrong with their muscle tissues, where their muscle tissues are breaking down or being damaged from something. Okay. So again, this is the review of the whole creatine phosphate route of acquiring and making ATP. So when you're resting, your resting muscle have a lot of ATP around, you're not using it, so let's not waste them. So it combines the phosphate group with a molecule of creatine to make creatine phosphate. And then now when you are actively working out again, now you need lots of ATP. So creatine kinase, as the enzyme, will then transfer that phosphate group back to ADP to give you ATP back. And these ATP can now be used for muscle contraction. So that's the whole part about how creatine phosphate is able to store ATP temporarily for us in forms of creatine phosphate. Okay, and it's a fast turnaround thing where we don't have to wait for glucose to go through that whole cycle of different steps. I mean, just glycolysis itself has 10 steps in order to make 2 ATP molecule, and then you have to go through citric acid cycle and electron transport chain to make 30 ATP. So, this is just a quick turnaround way so we don't end up wasting the ATP that's being generated while the muscles are resting. Okay. And on the, while we're talking about creatine kinase and clinical application, I actually found this very interesting case. Okay, this is actually a clinical case from a documentary show called Diagnosis on Netflix. It goes, I think when I was watching, I was like, wow, this is a clinical application standpoint from the, where we talk about creatine kinase in the muscle chapter. It is a very excellent, good example of how presence of creatine kinase in a patient's blood gave some clues about her metabolic condition. In this case, her metabolic condition, pretty much took away her normal life because of this very rare genetic disease that I'll get to at the end of the story. But this story begins with a 23-year-old woman who experienced severe pain to the point of being immobilized, and someone actually have to carry her every time she does strenuous exercise. And these pain can radiate from her legs all the way up to her cheeks, okay? And the 2 symptoms they saw in her was coffee brown urine and that her creatine kinase values were off the charts from her blood, which is a good indication that her muscle fibers are breaking down, but yet muscles should not be breaking down just from, for example, hiking a few miles. So what they see is that every time she does a little bit more strenuous type of exercise or activities, she ends up with these episodes of pain, coffee brown urine, and in her blood, her CK values are, creatine kinase values are really, really high. Okay, so something's going on where her muscle fibers starts breaking down and getting damaged and destroyed every time she is working out. And so by the end of the episode, they were able to find out that she has a very rare genetic disorder called CPT2 deficiency. This is where her body is unable to metabolize fat for energy. In a normal metabolism route, our bodies will utilize glucose first as our first source of energy. When glucose runs low, then it goes to lipid metabolism. And when lipid also runs low, the last resort will be amino acids from our muscles' protein filaments, which is something that we will talk about in the next semester in the metabolism chapter. All those different metabolism routes our bodies can take for energy. Now, rarely do our bodies turn to proteins as a source of energy, usually only if we were in a long-term starvation situation, like in a third world country where we're starving because there's no food out there. That's when and we haven't eaten for days, okay? So that's when we are using our last resort for energy, which is our amino acids within the muscles, like I was talking about, actin and myosin. So in this woman's case, somehow her body skips over that lipid metabolism part, and when her body goes low in carbohydrates or glucose, it goes straight for her proteins in her muscles, and hence her muscle tissues gets destroyed for energy, which then releases what's called myoglobin, some form of hemoglobin, a different form of hemoglobin that's specifically for muscle fibers and muscle cells. Because for muscles, it's so critical for them to have enough oxygen for contraction, for example, like your heart. Okay, don't forget about those other types of muscle fibers that also needs energy. Okay, not just skeletal muscle, but cardiac muscle, smooth muscle, all of those muscles needs ATP constant supply as well, and oxygen. So that's why you have myoglobin there to supply in a sort of acting like a last resort of oxygen. So when Your muscle fiber breaks down. Your myoglobin also gets released, and they get processed and released by the kidneys into your urine and result in her urine turning brown, because myoglobin is actually a much smaller molecule than hemoglobin, and that's why they can get processed into the urine, but it is still a type of oxygen binding protein which can turn brown when it is exposed to the air and no longer functioning in your blood. So this is why her urine was turning brown. So sometimes in clinical diagnosis is not easy because some symptoms can overlap between diseases, and one can only do a search and check kind of deal, via diagnostic tests. And even though we think, well, CK values off the chart, everybody would have thought it was a muscle degeneration thing, but it can be from all different types of diseases. Different types of metabolism diseases. For 9 years, she was tested for lupus, multiple sclerosis, because she was just experiencing pain that radiates up her body. So some people thought it was a nervous system related issue or a blood related issue because of her coffee brown urine. That's why someone thought it was lupus. But of course, all of those things came back as negative. It wasn't until she made a post to Dr. Lisa Sanders' column in the New York Times, which was what the Netflix documentary show was on. It was on her New York Times column with the more interesting cases she went through. So the whole point of that New York Times column is attract multiple ideas of diagnosis from physicians all over the globe that can narrow down to a diagnosis that may have, something that we kind of overlook, but someone else gave the idea, hey, maybe it's this, why don't you check it out? And then that's the way with rare diseases goes, is that sometimes within one doctor, one hospital, you may run out of ideas. And you may need other people's inputs. Maybe someone has treated someone with similar symptoms somewhere else on the other side of the globe, and they know, hey, that symptom sounds like my patient. So I thought it was an excellent idea for her to start this column, and it was very interesting, this whole documentary series. I watched every single episode, and for this lady, she was able to narrow down to this rare CPT2 rare disorder. Now, and treatment was very simple. She only needed a change in diet because it is a genetic disorder, so we don't have a permanent cure for genetic disorder. But at least there are some things you can do to avoid her body from using muscles as a form of energy resource. So what she does is she would actually avoid fat consumption in her diet and include high amount of carbohydrates in her body, which then she can continue to use for glucose as energy. Now, so the body will then, use whatever carbohydrate molecule she have in the body instead of targeting and starts breaking down her muscles and using amino acids as a form of energy instead. Okay, so now that we have a diagnosis and know a way to treat her, she's able to avoid those painful episodes and all those hospital visits that was interrupting her daily life. For 9 years, she was in and out of the hospital every 3 months, not knowing the reason why or what her diagnosis was. So, this was the end of her story, but and with CPT2 diseases, and the degree of severity can differ. In severe CPT2 cases, if left untreated, this can actually result in kidney failure, heart weakness, which can lead to early death, or liver problems that can lead to hypoglycemia, not enough glucose in your blood, and that can cause brain damage because your brain is the other than muscles. Neurons are the top priority for glucose, because the fact that we can send those action potentials down, we can propagate electronic signal for doing all those things our body does for us, is because our nervous system is working properly. If our brain doesn't have enough oxygen or glucose, it shuts down. Okay. So that's why when you don't have enough glucose, it's bad news for your brain. In a sense, it will lead to death pretty soon. So, In her case, she was lucky enough that probably it wasn't too serious of a case for her in terms of the CPT2 mutation that she had. But it was a very interesting case as we talk about creatine kinase and also metabolism routes. Okay, so the other two routes of muscle metabolism for energy is the normal cellular respiration process that you have to learn about in general biology. The anaerobic glycolysis is followed by anaerobic metabolism of citric acid cycle and the electron transport chain. In glycolysis, one glucose molecule gets broken down into two molecules of pyruvate with a net gain of 2 ATP molecules. Net gain meaning that it actually makes 4, but you spend 2 on the process of glycolysis. So that's why you end up with a net gain of 2 ATP molecules. For muscle fibers, the source of glucose will come from glycogen reserves, which are these branch polysaccharides that stores in your liver and your muscle cells, and they can be used for in times when you have already burned out all the glucose circulating around your blood. Then you go to your glycogen reserves, break down those glycogen into simple single monomers of glucose, and you can burn them for energy. And usually glycogen is stored within muscle cells within their sarcoplasm, where they're readily available for making of ATP, and ATP then can travel towards your travel towards your sarcoplasmic reticulum or for the release of myosin heads. All right. So pyruvate then will be converted to acetyl-CoA, which is the rest of the step going into the aerobic metabolism part of cellular respiration, which is your citric acid cycle and your electron transport chain. Okay, so the acetyl-CoA will go through the citric acid cycle right here, and it will pass electrons to your electron carriers, NAD and FAD, all that stuff. Then NADH and FADH goes on to your electron transport chain within the inner membrane of the mitochondria. It drops off the electrons, and then which causes those electron transport chain proteins, those coenzymes, to pump hydrogen ions into the mitochondria's inner membrane space, which then goes through ATP synthase to produce ATP. That's the whole process of what's happening within the aerobic respiration. And for one molecule of pyruvate, you generate about 15 molecules of ATP. So for a whole glucose molecule, you generate about 30 ATP. Again, this number changes depending which textbook you're looking at and which specific cells they're looking at. But 30-ish ATP is the right amount. It can vary from 30 to 38 depending on what the situation is. But definitely not, overshooting by 50 or going down but to the 15 level, somewhere in the 30s is what we usually see in the amount of ATP that is generated per glucose molecule. Now, a resting skeletal muscle fiber will rely on aerobic metabolism of fatty acids to generate ATP, but when the muscles are contracting, pyruvate would get broken down instead. Now, when a skeletal muscles is resting, you are not in high demand of very rapid usage of ATP. So that's why you then rely on slower form of metabolism. But when muscles are contracting, then you need very quick and massive amount of ATP supply, and that's why then they would use pyruvate instead and go through the aerobic metabolism for that quicker supply of ATP to your muscle fibers. We'll learn in second semester of AMP2 when we go into the metabolism chapter how that lipid metabolism actually is a very, very slow progress compared to glycolysis. So even though technically speaking lipid molecules is able to provide us with more molecules of ATP, but because it is a lot more cumbersome in terms of the whole process involved, your body still, first thing it goes towards for energy is glucose and carbohydrates. Okay, so here's a different metabolism pathway for a muscle fiber under various activity levels. So in a resting muscle fiber, your ATP demand is low. How about your oxygen level is good? You have enough oxygen around because you're not really doing anything. And ATP can be converted, can be converted from glycogen, or you can have storage of creatine phosphate, because this glucose is not actively being converted into ATP. So you have all these extra glucose around in your blood, which can be stored as glycogen within the muscle fiber itself. And if there's a lot of ATP hanging around not being used, let's not waste them. So they are stored as creatine phosphate. Now you decided, after sitting down for a while, okay, let's go for a walk outside. So that's an example of moderate activity. So the ATP demand now has increased, but not too much, not to a crazy level. Oxygen level in the muscle fiber itself, there's still enough. You're just doing some moderate activity. Now, glycogen reserve probably has now gone lower, because now you're using up your blood glucose for production of ATP, which is then used to support muscle contraction in the myofibrils. So when there's lower glucose molecule, there's a homeostatic effect to break down those glycogen for extra glucose that can now go through the process of aerobic respiration for making more ATP. There is an alternate route with fatty acid for lipid metabolism as well. That works too. But no ATP reserves currently, because all of them are being used for powering contraction. So all those all those creatine phosphate you have just stored, which now gets converted into ATP very, very quickly to supply your walk around the neighborhood. Now, let's say you decided, hey, instead of walking, let's go for a run, okay, a full out run. So now you are at peak activity of the muscle fibers. Your ATP demand now is enormous because you're running. And oxygen, probably not enough, because you're using those ATP so fast that you're doing tons of aerobic respiration, using up all those oxygen as your last electron carrier in your electron transport chain, your last electron acceptor within your electron transport chain. So now you're using up a lot of oxygen, but yet you're still demanding more. So there's not enough oxygen at the moment. So pyruvate also gets built up, because even though you're going through glycolysis very quickly, because glycolysis doesn't require oxygen, but your citric acid cycle and electron transport chain requires oxygen. So all this pyruvate is just waiting around for your electron transport chain to do its job, but it can't go any faster because you're running low in oxygen. So all this pyruvate, what do? Okay, so they can't just hang around, because then that would you need to actually free up your NAD and FADH2 and all that stuff too, for picking up more electrons. So that's why your body then shunts to lactic acid fermentation, okay, which is a type of anaerobic respiration that doesn't require your Krebs citric acid cycle and your electron transport chain to generate ATP. Again, you generate less ATP, but at least it's a way to generate something, and also free up your NADH and FADH2 to pick up more electrons for electron transport chain to work properly. You can't have them all held up by not being able to go through the electron transport chain. All right. But the caveat of this lactic acid fermentation or lactate fermentation is that you will have hydrogen ions build up, because lactic acid is an acid, okay? So that would decrease your blood pH, which leads to what we call as lactic acidosis. And we'll talk about this in the metabolism chapter in the next semester, going through metabolic acidosis versus respiratory acidosis. But this is an example of metabolic acidosis, decrease in blood pH due to metabolism stuff. So because your blood pH decreases, what happens is your muscle contraction will be inhibited at this point, and that's when your muscle will fatigue. So that's why I said our body can't rely on lactate fermentation for too long, because it actually builds up lactic acid in your body, and hence that is not going to last long, okay, because it changes and brings down your blood pH, and then that's when your muscle will feel very, very tired. Okay, this is when we're running, and all of a sudden they're like, man, we can't go on anymore. My muscles are hurting. My muscles are tired. Well, that muscle fatigue is due to that lactic acid buildup from being overactive muscles. All right, so what happens to all those lactic acid buildup during strenuous exercise? Okay, so lactate would actually diffuse out of muscle fibers because that's where they're generated, going through all that anaerobic respiration, and they will go into your bloodstream and continues even after exertion has ended because you need to catch up back on your normal cycle of metabolism. So once it goes into your blood, your liver will then absorb lactate. So this is that whole range of things your liver does for you, not just detoxification and generating proteins, but also in, the storage of glycogen and all that stuff. But it also turns lactate back into pyruvate, which then can be used to generate ATP or turn back into glucose. That's what your liver can do. Okay, gluconeogenesis, that is able to make glucose from different parts of the glucose molecule. Okay, we'll talk about, again, something we'll go into and talk about in the next semester. Don't worry about it for now, but that's what liver can do, and that's how it can neutralize your lactic acid or lactate in your blood, is by doing this part of it. Okay, and this part that neutralize your lactate is called the Cori cycle. Okay, so don't forget Cori cycle. Glycolysis, okay, for turning pyruvate into ATP, or lactic acid fermentation for anaerobic respiration when we don't have electron transport chain available, and Cori cycle for neutralizing lactate back into pyruvate that can either turn into ATP or back to glucose in the liver. And if there's too much glucose, then it will store them as glycogen. All right. And because ultimately you still need to use oxygen to burn all those glucose molecules for ATP. And remember, when you're at peak activity, you ran your body out of oxygen. And so you are sort of, putting a loan down for oxygen. Hey, we don't have enough oxygen right now, but hey, let's do this other stuff. But that doesn't mean you don't have to pay back those oxygen back to your body. Okay, it doesn't work that way. Kind of like how money works. Okay, so this is what we mean by paying your oxygen debt back, which is the amount of oxygen required to restore your normal pre-exertion condition, which this is called excess post-exercise oxygen consumption, okay? EPOC, okay? EPOC, excess post-exercise oxygen consumption. So that this explains why even after we've stopped exercising, you're tired, you stop, but you're like, you're still breathing very hard after the exercise, is because you are paying your oxygen debt back. You've stopped your strenuous exercise, you stop your massive demand for ATP, but you have all this Cori cycle that you have to do afterwards to get rid of those lactate, and that's why you breathe heavily to help process those extra stuff. Okay, so that's why you breathe heavily even after you stop exercising. Now, hormones actually play a role in muscle metabolism as well. For example, growth hormone from the pituitary gland and testosterone can actually stimulate synthesis of the contractile proteins, okay, actin and myosin. Again, this is only testosterone that can stimulate that process more often, and that's why males have a lot easier time building muscles, really large muscles compared to females because of that. And but growth hormone and testosterone is able to cause an enlargement of skeletal muscles based on demand. The thyroid hormones also help in elevating rate of energy consumption, because thyroid hormones, we're not talking about calcitonin, but we're talking about thyroxine, T3 and T4, regulate metabolism in the body. So that's why they can also have a role in playing the rate of energy consumption in resting and active skeletal muscles as well. So these are just a couple things that can affect your rate of metabolism, muscle metabolism. And while we are on the topic of muscle metabolism, different types of muscle fibers are actually more adapted to do aerobic respiration versus anaerobic respiration. Slow fibers, for example, are specialized in ways that enable for long-term aerobic respiration. Okay, they are able to continue contracting for a long time because they have lots of mitochondria around to do lots of aerobic respiration. Okay, so slow fibers are specialized for doing a lot of aerobic respiration. And they also, because they have to do a lot of aerobic respiration, they have more extensive work of capillaries, which enables them to have more oxygen for that aerobic respiration. And like I was talking about, they contain a special hemoglobin form called myoglobin. So that's why it starts with myo, because they are specialized to be in the muscle cells for providing more oxygen other than relying on hemoglobin. So that's why muscles that are slow fibers usually looks a lot more dark red in color. So an example of slow fiber is the one in your gastrocnemius, for example, which allows us to do more long-term running, walking, and things like that without feeling super muscle fatigue just from, walking, doing a walk around the neighborhood, for example. Okay. All right. So slow fibers, the other thing about them under the microscope is they usually have a smaller diameter and takes about 3 times as much to reach peak tension after stimulation, 3 times the amount of time to reach peak tension. So that's why when we were comparing the twitch time duration between your eye muscle, gastrocnemius, and soleus, that's why there was such a big difference in the time period of the twitch, is because it depends whether they are slow fiber or a fast muscle fiber. Slow fibers, they're there for that long haul of aerobic metabolism to continuously provide ATP. For example, like I was talking about in the leg muscle that allows us to walk and run. Now, the other type of fibers is called fast fibers. So fast fibers are larger in diameter, very different from slow fibers. Then they can reach peak tension in less than 0.01 seconds after stimulation. That's why they're called fast fibers. They have those fast twitches. They have fewer mitochondria, though, because they're not meant to do prolonged activity per se. Their contraction usually requires more ATP too. So that's why if the activity somehow becomes longer for these fast fibers, they will be more reliant on anaerobic respiration. They're not built for aerobic respiration. Okay, so fast fibers is for anaerobic respiration purposes. Because they are not meant for that long haul of needing ATP consistently. They're for that fast movement. Okay. So, an example of fast fibers are those muscles that control the eye movement, like I was talking about, which rarely are they doing prolonged activity. I mean, you don't slowly move your eyes to the left or slowly move your eyes to the right. You kind of glance around very quickly, blink here and there. These are very quick movements in a very short time frame. So that's why our eye muscles have a very, very short twitch duration. Now, our muscle size can also differ with the amount of usage as well. The diameter of our muscle fibers can increase with repeating cycles of stimulation that produce almost maximal tension consistently. This will result in what we call muscle hypertrophy. So that is why they say if you want to build muscles, you have to make sure you're not, if you continue, let's say you're a really built person, you can't just continue doing that 5-pound dumbbell all the time. Hey, I'm doing muscle hypertrophy with this 5-pounder dumbbell, because you are not using the maximal tension of your muscle. In order to trigger your muscles to think, hey, we're doing we're carrying these heavy stuff and we need to maximize our tension all the time. So in order for us to continue to supply our body's needs for that constant heavy tension that's needed for this 50-pound dumbbell, we need to increase in size. We need to get a bigger muscle going in order to be able to build that. So that's why you have, those bodybuilders gets heavier and heavier in those weightlifting, sessions, is because that's what build muscles. Now, on the other hand, if we don't use our muscles, muscle fibers can decrease in diameter, muscle tone, and even muscle power. And we call this condition muscle atrophy. So, for example, in case where individuals resulted, because of some, maybe accident or something that resulted in long-term paralysis, okay, and sometimes it can differ in reason why someone can have paralysis, maybe spinal cord injuries, or from viral attack, especially polio, for example. But paralysis means which means you can't move. Okay, so if you can't move your legs, for example, you gradually lose muscle tone and size in your legs. So this is why when you see a person that's chronically on a wheelchair, you see that their legs are very skinny, is because they haven't used them. And if you don't use them, you lose muscles. They have undergone muscle atrophy. And in those with poliovirus infection, this particular virus attacks motor neurons in the spinal cord and brain specifically, and which can lead to muscular paralysis and consequently muscular atrophy as well. Now, for extreme atrophy, muscle functions can actually be permanently lost. Okay, so that's why physical therapy is very critical for those who have temporarily lost their ability to move and walk and need to get back on track. So, for example, you have a fracture on one of your ankle or your leg, and hence you were on crutches and a cast for months, and then so you haven't been using that muscle for months. And when you're off the cast, that's why even though sometimes it can be painful for some people, you still have to do that physical therapy to get your muscle going again, so it knows, hey, I'm being used, so I need to grow and get bigger. Okay, so that's the part of telling your muscle that there is a need, and hence they will build up their strength to get you back to normal daily movement. Okay. Now, aging also has an effect on our muscle tissues too. So, for example, when we age, skeletal muscle fiber actually will become smaller in diameter. They also become less elastic, and they have less tolerance for exercise. And the ability to recover from muscular injuries also decreases as we age. Now, as we have mentioned about, talking about complete tetanus, our muscles can get tired and go through fatigue when we're talking about, maximizing our muscle tension. So when our muscles go fatigue, it is correlated with a couple of different things. Okay, so for example, you have depleted your metabolic reserves, like your glycogen and glucose and all that stuff within the muscle fiber. So that's why you feel muscle fatigue, and you feel like you got to stop. Okay, after you've been running, you're like, no, I can't go on anymore. It's because you're running low in metabolic reserves. The other thing that can happen is maybe there's a damage to your sarcolemma or the sarcoplasmic reticulum that is not the function. The muscle fiber itself is not functioning correctly. That decline in pH value we were talking about earlier, because a more acidic condition can alter enzyme activities. And lastly, the sense of weariness and reduction in the desire to continue the activity is the last thing that tells you, okay, I gotta stop because I can't go on anymore. Okay, so that's why they talk about, when I was in ROTC back in high school, they tell us my sergeant always tell me, mind over matter. Okay, so sometimes you just have to keep going and tell yourself you got to keep going, keep putting your muscles at that maximal tension, and then finally you will be able to build longer and longer stamina. Now, I'm not saying to continue if you're feeling faint or feeling really bad and like heat stroke and all that stuff. When your body is really at that edge, okay? But I'm saying that, just little bit sense of weariness, keep going, keep trying, okay, is what, helps build your muscles, all right? but not, in the case if you're going to faint or something, okay? All right. So for example, marathon runners will see muscle fatigue when all of their glycogen, fatty acids, and protein reserves are depleted because they're going in for that long-term, long haul of continuous usage of ATP. Sprinter, on the other hand, experience fatigue from lactic acidosis. Okay, so this is, for example, how typical marathon runner looks like versus a sprinter. And you can see the difference in their muscle size is because one is relying more on aerobic respiration. The other is relying on that burst of power from anaerobic respiration. And because you need that burst of power very, very quickly, very, very massively in a short amount of time, that's why they need to really have huge muscles in order to power that ATP for that short amount of time. And hence, for athletes specializing in different types of sports, one may build different kind of muscle metabolism endurance. For example, if one tends to increase anaerobic endurance as in power sprinting, athletes will need to perform frequent, brief, intensive workouts that stimulate muscle hypertrophy that will enhance glycogen breakdown, okay, such as weightlifting, resistance training, and all that stuff to build your muscles, get bigger, larger muscles. Now, to increase aerobic endurance, if one wants to increase that, then training would involve sustained low levels of muscular activity, such as long distance jogging, long distance swimming, exercise that don't really require that peak tension production, but you're training your cells to keep doing that long haul of making ATP, so your cells will start making more, having more mitochondria to apply, to help you supply that demand, that constant mid-range demand of ATP. Okay. All right. But technically speaking, if most athletes kind of do both aerobic and aerobic endurance training, okay, this is called cross training, where we are able to do a bit of both so that they get both endurance and that burst of power at the same time. All right, and then we will end this chapter by going over briefly how cardiac muscles and smooth muscles are different from skeletal muscles in terms of microscopic structures. Now, cardiac muscles also contain myofibrils and also align sarcomeres, just like skeletal muscles. They're closer in appearance to skeletal muscle fibers than smooth muscle, that's for sure. That's why we still see striation like this all over the cardiac muscles, but there is something special called intercalated disc that we'll talk about. So the difference is that cardiac muscles usually contain only one single nucleus, whereas skeletal muscles, you can have multiple. Cardiac muscles are also not completely long cylindrical shape, but they are branched and interwoven for stimulating that rhythmic muscle contraction of the heart that we'll talk about in the next semester. There's also their T-tubules looks a little different. They're usually short and no triads, which means they don't have a terminal cisternae. So that's why you see just these meshes going straight into the T-tubule versus that enlarged areas we saw in skeletal muscle, because it doesn't have that terminal cisternae within their sarcoplasmic reticulum. Okay, they have intercalated discs for action potential to move very quickly between your cardiac muscle cells. For cardiac muscle cells, you can't fatigue, okay? If a muscle cell in the heart fatigues, the heart stops pumping and you die, okay? So this is why it is even more critical for the heart to be able to access and regulate and spread that action potential very quickly between the cells. Okay, skeletal muscles, we can stop and be tired, but muscle in the heart cannot be tired. And that's why for a heart to pump, okay, in that rhythmic average heartbeat that we have, it doesn't require neurostimulation. Okay, we'll talk about this towards the end of the chapter when we get to autonomic nervous system. Because there's a lot of things that's happening in our body that doesn't constantly require our brain to stimulate for it to work. Okay, so heart, for example, if every heartbeat requires your brain to do something, then your brain will not have any other capacity to do other stuff for you. The thinking, the critical thinking, the decision making, all that stuff that your brain has to do. So something that we have to do every, all the time, consistently, every couple, every second, okay, millisecond even, that, require neurostimulation. Okay, this is called automaticity. And your heart cells actually have a little area within the heart called the sinoatrial node, we'll talk about in the next semester, that has these cells called pacemaker cells. That's what determines the timing of the contraction and sends out an electrical stimulus that goes through the rest of the cardiac muscles for them to have that, constant heart rate. For heart, the contraction actually 10 times as long. So, you don't see your heart doing dip, 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 dip. It doesn't do that. It doesn't do dip, 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 dip like that. Okay, like your skeletal muscles. If you were to quickly swerve away from something, or if you touch something hot and your muscle pulls back really quickly, your heart doesn't do that kind of contraction. Your heart is slowly pumping, squeezing on the blood, pump, atria, ventricle. They're doing slow contraction compared to our skeletal muscles. So that's why they don't readily fatigue, because their twitches will be a lot longer. In addition to releasing calcium from the sarcoplasmic reticulum, our heart cannot not have enough calcium, okay, or else the contraction stops, and we can't afford to have our contraction stopped in the heart. So calcium ions can also flow in through the sarcolemma with an action potential. That is something different from skeletal muscle fibers compared with the heart. Just because for the heart, we cannot afford to not have enough calcium ions. So this is why we talked about in the bone chapter, why we need to regulate our calcium ion with such stringent, parameters, is because our heart cannot afford to not have enough blood calcium. Okay, so for smooth muscle, they're almost completely different from skeletal muscles. Their cell, for example, is spindle shape versus that cylindrical shape. Okay, and they form sheets, bundles, or sheath around tissues. They're just shaped very differently, and that's why microscopically speaking, they are almost completely different. Now, functionally, they're also different. Okay, but let me finish with the structural differences first. Again, they're one smooth muscle cell only have one nucleus. That's different from the skeletal muscles. Now, they actually don't even have any T tubules at all. No T tubules. And their sarcoplasmic reticulum forms a loose network throughout the sarcoplasm. They don't have myofibrils or sarcomeres compared to skeletal muscle. Their thick filaments are scattered all over the place instead of in a nice organized thing. We talked about the I-band, A-band, etc. There's none of that in smooth muscle. The thin filaments are attached to these blue dots you're seeing here called dense bodies. And because it is not in a straight line, smooth muscle fiber their contraction doesn't result in that linear contraction. They actually result in twists. Okay, so this is a relaxed, for example, smooth muscle, and this is a contracted smooth muscle. Because of how their filaments are oriented, they have that twisting motion every time they squeeze, which makes sense because then these are usually surrounding your digestive system, for example, your stomach, your intestine. They're squeezing and moving food along or blending the food along, so they need those twists of muscle. They're not really for that linearity of movement of muscle, okay? And also, they don't have tendons or aponeurosis. Functionally, there's influx of calcium ions still for activation of smooth muscle, but they bind to a different protein called calmodulin. Okay, so there's no troponin. Okay, there's no troponin, there's no tropomyosin. So calmodulin is what calcium binds to in smooth muscle, and they activate myosin light chain kinase instead, and that's when myosin heads will then bind to actin. So the thin filament components are also very different for smooth muscles. And the last thing about smooth muscles that they actually, in terms of innervation, smooth muscles can have two different categories of innervation. One's called multi-unit smooth muscle, and the other is called visceral smooth muscle. With multi-unit smooth muscle regions, one muscle cell may connect to more than one motor neuron. Okay, now that's different because we're talking about in skeletal muscle only one neuron or for one muscle cell. Okay, but for smooth muscle, one muscle cell can connect more than one motor neuron. So several motor neurons can innervate one smooth muscle. For example, those in the iris of the eye for regulation of pupil diameter, or those erector pili in the skin we talked about in the integumentary system. Those are all smooth muscles that we have no control over, but is automatically set by your nervous system. Okay, so that's why those is known as the multi-unit smooth muscle. The other category of smooth muscle are your visceral smooth muscle, and those are actually not innervated by motor neurons. Okay, just like what we're talking about with cardiac muscle, they don't need neurostimulation to do their job. The electrical impulse would actually travel through gap junctions in wave-like movement through the entire layer, triggered by, again, some pace setter cells, just like your cardiac muscles. Okay. For example, those smooth muscles within the digestive tract or the urinary bladder, you can't have your brain having to interrupt every time your feces are moving along section by section in your large intestine. It should be an automatic thing your intestine does as it senses the pressure coming through with the feces moving down the length of the large intestine. All right, and so this wraps up a very long chapter 10 on muscle tissues. That's why I give you 2 days to go over muscles. Okay, so let's go through the practice questions and we will be done. Okay, how are epimysium, perimysium, and endomysium interrelated? They are interconnective tissue that blend with tendons to provide firm attachment to the bone. Okay, they don't all contain capillaries. They don't all contain satellite cells. Why does skeletal muscle appear striated when viewed through a microscope? We talked about this immensely. So Z-lines and myosin filaments align within muscle fiber. So myosin filaments are those dark areas, and then active filaments are the thin areas. Because of the protein structure, that's why they appear dark and light. Okay, myosin are thicker and bigger, whereas actin are thinner. Okay, nothing to do with staining. Okay, the A-band comprises which of the following subunits? Comprises which of the following subunits? Your A-band has the M-line, the H-band, which is that zone without the actin filament, and the zone of overlap. In what way does the appearance of a sarcomere change during contraction of a skeletal muscle fiber? This is that sliding filament I was talking about, okay? So the Z lines would move closer together. The I and H bands would also get smaller. Okay, so zones of overlap in choice A here is true, but H band does not get wider. It gets smaller. And A bands, remember I said that A bands do not change in length, whether you're resting or contracting muscles. How would a drug that blocks acetylcholine release affect muscle contraction? Well, acetylcholine is essential for regulating that action potential down in the sarcolemma of the muscle fiber. If you block acetylcholine release, you have no muscle contraction. So it will be prevented. You won't even have a muscle contraction. It's not just slow down. You're completely eliminating it. Okay? Because there's no signal for the muscle fiber to contract. Which of the following describes a neuromuscular junction? It is that special intercellular connection between the axon branches or axon terminals with the skeletal muscle fiber. As an axon enters a muscle, it branches into a number of axon terminal, each of which makes contact with a single muscle fiber. The portion of the sarcolemma in contact with the axon terminal is called the motor end plate. Okay, this is talking about sarcolemma, which is the muscle fiber side. What do you have? Remember those curvatures I was talking about? That makes up that synaptic cleft where the axon terminal goes into. So this little part of the sarcolemma is known as the motor end plate. Okay, what is the name of the enzyme that degrades ACH? Acetylcholinesterase. Very easy to remember. Starts with acetylcholine to begin with. Esterase. Okay, just think of it as acetylcholine elimination or something. Okay, so you can tell that it is for digestion or degrading away your acetylcholine. Which activity would be more likely to create an oxygen debt? Swimming laps or lifting weights? Okay, it would be lifting weights. Okay, oxygen debt, anaerobic respiration. Okay, which means you're doing something very, very powerful that requires massive amount of ATP. So that's why lifting weights will create oxygen debt more so than just doing, average range tension activity required for swimming laps. What statement below is false with regard to contraction of skeletal muscles? Through concentric contractions, muscle does not change in length. That is not true. Concentric means it gets shorter. Eccentric means it gets longer. Think of eccentric for elongation. Concentric, something more concentrated, so shorter, okay? And isometric is when it doesn't change in length. Which of the following describe the characteristics of slow muscle fiber? They have smaller diameter, rich in myoglobin. That's why they are more red, dark red, and very vascular for that long-term aerobic respiration. How would severing the tendon attached to a muscle affect the muscle's ability to move a body part? No movement is possible because once you cut off a tendon, your muscle can contract all it wants, but it can't move the bone. No movement of the bone, no muscle, no movement, okay? A motor unit is or are what, and you would need a small motor unit to control movement of the fingers. In what way? Okay, so a motor unit is where the muscle fibers are controlled by a single motor neuron, and one motor neuron can control multiple muscle fibers. And when you need to control movement of fingers, you need a small motor unit because you need that precise control, that fine motor movement by your fingers. Where would you expect the greatest concentration of calcium ion to be in a resting skeletal muscle? Well, it's resting. It's not during contraction. So where should they be? Within the terminal cisternae of the sarcoplasmic reticulum, nicely stored away. And then only when your muscle need to contract, then they get that action potential coming through from the T-tubules, release the calcium ion into the sarcoplasm. What predominate in the large leg muscles of someone who excels at endurance activity, such as cycling? This is because, slow fibers, because this is endurance we're talking about. They have to have that long haul of ATP provision, and they have extensive capillary networks and lots more myoglobin. Why is an increased level of creatine kinase in a blood test a diagnostic sign of muscle damage? Is because this enzyme is supposed to be inside your muscle fiber cells, not outside. So the fact that they're in the blood means there's something damaging the cells. The plasma membrane is leaking this creatine kinase into your blood. What would you expect to happen in a resting skeletal muscle if the sarcolemma suddenly became very permeable to calcium ion? Okay, so now you have too much calcium ion. What happens? You have that decreased ability to relax. Your muscle fibers are going to remain in contraction, and they cannot relax unless those calcium ions are able to be retrieved by your sarcoplasmic reticulum, or they're gone in another method, okay, by diffusing out of the cell or something, okay? Predict what would happen to muscles if a pesticides that inhibit acetylcholinesterase were present at the motor end plate. Oh, then we're in trouble because now you're having this enzyme, okay, that is supposed to be able to get rid of ACH, but now you don't have those enzymes, so your muscle will contract repeatedly. So it's kind of like, spraying pesticide on us and freezing our muscles up, okay? Cardiac muscle fibers are more sensitive than skeletal muscle fibers to which extracellular ion? Calcium. The only ion we've been really talking about immensely for muscle contraction. Sodium is only for that action potential. So sodium, potassium will be our key ion when we talk about chapter 12, okay? What structural characteristics distinguish cardiac muscle tissue from skeletal muscle tissue? Cardiac muscle cells they have intercalated discs, they have short T tubules, and no triads. Why would a sprinter experience muscle fatigue before a marathon runner? Because sprinting involves anaerobic endurance, which makes lactic acid, whereas running a marathon depends more on availability of substrate for aerobic respiration. Do you have enough carbohydrate for the running? Okay, and that's all I have for you in this chapter. I already, like I said, as you saw in my next slide, I actually already eliminated a couple slides from the first time I taught this course. So it's a really, really long chapter in your textbook with lots and lots of information. So that is why it is this long. Now, it's a difficult chapter. Make sure you spend the time on studying on this chapter. And the other thing is, what I see students having trouble with is confusing chapter 10 with chapter 12 stuff instead of making them into a full story. So don't confuse what calcium ion does in muscles with what calcium ion does in neurons. They do two very different things. Neurons don't contract, okay? But still, neurons require calcium to do something else, as we will talk about in chapter 12. So it's good to, write a lot of notes, draw a lot of diagrams for this chapter so you have it down solid, and then move on to chapter 12 for neurons. Okay, I'll see you in chapter 12.